tuning in to Planetterrian Daily, episode 130. It's July 24th, 2026. Let's see what the latest research is telling us. Single-cell mapping shows human hippocampal aging proceeds in distinct, non-linear stages with microglia replacement by blood-derived cells. Single-cell multi-omic profiling of the human hippocampus shows aging does not advance uniformly. Microglia populations turn over from blood-derived precursors rather than remaining embryonically derived throughout life. The same dataset documents progressive astrocyte loss. Researchers are now correlating these stage-specific signatures with cognitive scores in living donors. Longitudinal sampling in additional brain regions will test whether the nonlinear pattern holds elsewhere. From the cellular choreography of brain aging, we turn to a specific genetic factor that appears to slow similar processes. The ApoE2 variant reduces both disease-related and age-related brain pathology. Researchers examined genetic data alongside brain pathology markers in the study published on ScienceDaily. The protective effect stands out against the more commonly studied ApoE4 risk allele. This finding adds to ongoing work tracking how specific alleles modify late-life brain resilience. Follow-up studies will test whether the mechanism can be mimicked therapeutically. While genetic protection can slow brain aging, another study has identified variants that drive a very different health burden. Specific sequence changes markedly raise risk for hyperemesis gravidarum. Investigators compared genomes from affected individuals against population controls. The implicated loci point to hormone signaling pathways that become dysregulated early in gestation. The work supplies the first clear genetic architecture for a condition long viewed as purely environmental. Larger cohorts are now being assembled to confirm effect sizes across ancestries. From pregnancy biology, we move inside the cell to a metabolic enzyme now linked to broader lifespan extension. Inhibiting acid ceramidase alters ceramide metabolism in ways that reduce cellular stress accumulation. Animal models show measurable gains in tissue function and survival curves. The pathway intersects with known longevity regulators without requiring broad metabolic suppression. Human cell and organoid experiments are underway to assess translation. Next steps include dose-finding studies in aged primates. Longevity pathways often intersect with mitochondrial function. University of Cologne researchers mapped the link between disulfide bond formation and heme production inside mitochondria. The process couples oxidative folding of imported proteins to the availability of heme for respiratory chain complexes. Disruption of either step impairs ATP generation and triggers compensatory stress responses. The mechanism was demonstrated across multiple cell types using targeted knockdowns. Future work will examine whether boosting folding capacity can offset age-related mitochondrial decline. Most people picture mitochondria as simple power plants that burn fuel and release ATP on demand. Right now, inside your cells, the same compartment is running a tightly coupled quality control system that decides whether heme gets made at all. When incoming proteins fail to fold correctly and form their stabilizing disulfide bonds, heme synthesis slows, starving the electron transport chain of essential cofactors. A single heme molecule participates in roughly 10 to the 12th electron transfers over its lifetime inside a typical cell. Interventions aimed only at boosting mitochondrial biogenesis may underperform if they ignore upstream folding capacity. Watch for upcoming trials that combine folding enhancers with existing metabolic therapies. New spatial tools are also advancing proteomics. Full-scope-seq merges stereo-seq spatial capture with long-read sequencing. The method maps transcript isoforms across large tissue areas while retaining cellular identity. Application to macaque brain slices revealed cell type-specific splicing patterns not detectable by short-read approaches. The dataset covers multiple cortical and subcortical regions in one experiment. Additional primate and human samples are scheduled for the same pipeline. A Python package processes highly multiplexed fluorescence datasets from start to quantitative output. It integrates segmentation, neighborhood analysis, and spatial statistics in a single interoperable workflow. Developers validated the tools on human and mouse tissue sections containing dozens of markers. The release lowers the barrier for labs without dedicated imaging informatics teams. Planned extensions include support for emerging cyclic immunofluorescence protocols. University of East Anglia, Durham University, and University of Cape Town teams compiled the first comprehensive inventory of coastal World Heritage locations below 20 meters elevation. The digital map integrates elevation, erosion, and flooding projections. It supplies planners with site-specific exposure rankings for the first time. The resource is openly available for adaptation modeling. Updates will incorporate new satellite-derived elevation data as they become available. Murdoch University and Wallach University researchers designed biodegradable structures that anchor seedlings against currents. Early field trials show improved survival compared with traditional planting methods. The material degrades without releasing persistent microplastics into the sediment. Scaling trials are planned across additional Thai coastal sites. Long-term monitoring will track meadow expansion and carbon sequestration rates. The Kuhlman Island colony produced roughly 69% fewer chicks in 2025 than the prior year. Researchers traced the decline to a large iceberg that blocked access to foraging grounds from July 2025 through January 2026. The event occurred despite broader sea ice conditions that appeared favorable. The finding illustrates how localized ice dynamics can override regional averages. Continued satellite tracking will test whether similar blockages recur in future seasons. UCL-led analysis shows that shifting training age by even 1 year would reduce plastic waste substantially. The study models both behavioral interventions and emerging recycling technologies. Current global diaper disposal contributes millions of tons of non-degradable material annually. Pilot programs in several countries are testing combined education and collection schemes. Life cycle assessments will quantify net environmental gains. 2 decades of temperature, salinity, and pressure profiles were processed to map circulation features at unprecedented resolution. The approach identifies previously undetected transport pathways and mixing zones. Results align with but refine existing ocean models used for climate projections. The dataset remains publicly accessible for further refinement by other groups. Additional sensors on future floats could extend the same methods to biogeochemical variables. OpenAI's model produced a proof for the unit distance conjecture in May 2026 after decades of stalled progress. The result demonstrates that AI systems can now tackle problems previously considered out of reach for automated methods. Mathematicians are verifying the proof line by line while exploring extensions to related open questions. Additional benchmark problems are being prepared for the same systems. The newly examined manuscript contains candid reflections on sexuality, literature, and daily life. Archival analysis places the writing in the early 1950s, before Turing's prosecution. The story challenges the narrow public image of Turing as solely a codebreaker and theorist. Historians plan to release annotated excerpts alongside the original text. The find adds texture to ongoing reassessments of mid-century scientific culture. Experiments show that specific wall compositions increase neutron yield without raising input energy. The effect stems from altered plasma surface interactions that stabilize the reacting fuel. The finding matters for both energy research and neutron source applications in medicine and security. Multiple material candidates are now under systematic comparison. Scaled tests will determine whether the gains persist at reactor-relevant conditions. The chemistry proceeds inside living tissue without toxic catalysts or external triggers. It enables selective conversion of inert precursors into active compounds at tumor sites. Cell and animal data confirm reduced off-target effects compared with conventional activation methods. The approach broadens the design space for pro-drug strategies. Human safety studies are in planning stages. Next time, we'll be watching for results from the primate dose-finding studies on acid ceramidase inhibition. That's Planetterrian Daily for today. If you enjoyed this, a rating or review on Apple Podcasts or Spotify really helps new listeners find the show. I'm Patrick in Vancouver. Thanks for listening, and I'll see you tomorrow. And if you'd rather watch than listen, find us on YouTube at NERA Network. Links in the show notes. And before you go, this show is part of the NERA Network, a family of daily podcasts on tech, science, and markets. Each one a short, sharp briefing you can finish on the commute. If today earned its place in your feed, here's your next listen. SpaceX Daily, the daily companion for following SpaceX as a public company, from Starship to the stock ticker. Find every show free at neranetwork.com. The age of AI puts real builders on the phone with an AI host. Apply at neranetwork.com/age-of-AI apply. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.